Good morning to all my St. John's College of Pharmaceutical Sciences and Research, Kattappana, Kerala, 5th semester students. I would like to upload this class which I have taken on 16th July, Thursday, 2 to 3 p.m. Kindly listen to this audio presentation and make use of it if suppose you missed your classes on that day. This is about the pharmacognosy and the phytochemistry 2 in our syllabus, unit 1, and this is our first class of this semester. So let us see about the course outline which is given by PCI and also our university. According to that, in first chapter, it says to be covered all the metabolic pathways in higher plants and their determination also. And in that, we have to discuss about brief study of basic metabolic pathways and formation of different secondary metabolites through these pathways, especially ischemic acid pathway, acetate pathways, and amino acid pathways, and then study and utilization of radioactive isotopes in the investigation of biosynthetic studies or biogenetic studies. So now we are coming to the class, but we cannot cover all the things within this 1 hour lecture. At least we can cover the ischemic acid pathway and how it is applicable to produce aromatic amino acid in nature, in biosynthetic pathway. So now we should know about first what is a primary and secondary metabolites. So this is we might have come across, already studied in your previous classes, especially biochemistry. In that you might have studied about human biochemistry and what all the metabolic pathways which happens in human chemical industry or chemical factory. Like human body, it has many chemical reactions which take place. In that you might have studied about primary metabolites and secondary metabolites. In human or in animal, we have various metabolic pathways or the primary metabolites, and it is converted into secondary metabolites, and it may be excreted through urine or feces, and these are all the things which happen in the animals. And also it produces energy, and you might have very much clear about, your teacher might have told you to study about citric acid cycle, glycolysis, and how many ATPs are produced, energy is produced, and also you might have studied various definition about what is catabolism, what is anabolism. Like that you might have studied many words. So same thing we believe that, and also scientifically it is proved that even plants are also having life and living organism. So there also we have the same chemical factory and chemical reaction takes place inside the plant. And we know that plant produces their own food. Photosynthesis you might have studied in your smaller classes, which produce the polysaccharide starch, and which has been utilized by the plant for their survival. So, a primary metabolite always needed for any living organism for their survival. So, we have primary metabolites such that carbohydrates, proteins, fats, and nucleic acid. So, what do you mean by secondary metabolites? So, secondary metabolites, these primary metabolites are even converted into various metabolites. By means of joining them, it produces various secondary metabolites. Sometimes if you are taking some drugs, it also metabolized. Always it undergo biotransformation, metabolized to another compound, and it can be excreted. So this is what it happened in the animal. But in plants, there are many secondary metabolites in nature by our creator. It has been produced and it has been utilized by human being for various medicinal purposes. So we are more interested to know about secondary metabolites present in the plant sources, and also even animal sources, and even minerals, which comes under our drug use, medicinal drug use. But you see, the nature, we have secondary metabolites, various secondary metabolites like alkaloids, glycosides, saponin glycosides. There are many different glycosides, flavonoids, resins. So we have many secondary metabolites which has been utilized by human being for their medicinal uses. So here, where these secondary metabolites are produced by the plant, and as it is not having any excretory organ, so it is stored in the plant cells, and so that we are utilizing it. Various plants, sometimes the secondary metabolites concentrated in the leaf, sometimes it is present in the flower, roots, bark, and various parts of the plants. It has been taken. Sometimes it has been utilized like an exudates, which is obtained from the plants, is also utilized as an secondary metabolites or at a medicinal purpose, or even normal food, and everything we are utilizing this plant source. So now we are going to about this. And what do you mean, what is the difference between, because we are going to study about the metabolic pathways and we are going to study about biogenetic pathways. It has been given in the syllabus. So what is biogenetic pathway? What do you mean by biosynthetic pathway? What do you mean by synthetic pathway? If the student know these differences, it will be very easy for you to study this chapter. So synthetic pathway, you might have studied in your synthetic chemistry. Your teacher might have starting material and intermediate catalyst may be added, and you will make one final product and you will say this is the final product which I have obtained in the laboratory. So in laboratory you are taking some chemical minimum small initial compound for producing one final product. Reactant will give you a product with the help of, sometimes you may be adding some catalyst, and various procedures will be followed to produce the final product. So that is a synthetic pathway which is produced by the human being in the laboratory. So that is what even in the last class, last semesters, you might have studied about this in chemistry, and also artificially you are making even last pharmacognosy and phytochemistry 1, even I taught you regarding about the tissue cultures. That is also a modification which have been made by various modification which has been made by cellular or genetic studies, and we have made many types of plants, and hybrid varieties can also be produced by means of various cultures which can be done by tissue culture, plant tissue culture, animal tissue culture. There are many types of tissue cultures are there which we have come across in our previous semester. Now you should know about what is the difference between, what do you mean by biosynthetic pathway or biogenetic reactions or biogenetic study which we have come across, we are going to see now. So what is this? It is also same thing. Why it is called bio? Why it is called bio? It is naturally which is the reaction taking place inside the living organism. That is why we are using always bio, bio, bio. So bio transformation or genetic pathways, that means biogenetic pathways, that is, or biosynthetic pathways, which happens inside the plant by how the reaction takes place. Just imagine what you have studied in your biochemistry. You just remember. You can see that even I do remember what we studied in our smaller classes. That is, example, glycolysis means immediately you will remember a glucose molecule will be converted into pyruvic acid. And at the pyruvate will enter into citric acid cycle. And it will be converted into various form like aconitate, and citrate and fumarate, and then it comes to, and it is made into many forms, and it produces ATP and gives the energy for any type of work made by that particular human being or particular animal. So energies, ATPs, are produced by means of this metabolic pathways. So you have studied already what is glycolysis and how it has a parameter to give you and give you how it can produce energy, and it have various parameters, various cyclic, and what is happening there, what is responsible, what is the catalyst, how it is happening. Are you using any test tubes or anything? No, inside the living organism there are enzymes. These enzymes are responsible for conversion of one molecule to another molecule. So we are going to study about what all the enzyme responsible for this conversion and how you are getting a compound secondary metabolite. Because already we have studied there are alkaloids, glycosides, flavonoids, tannins, and many natural compounds are there. And these have various chemical structures. And how these structures are formed? Is it they exist from their seed itself? No. When they become seed, when they become a small plant and then become a tree or a shrub or whatever may be the size of the plant, and after the development only it is developing. How this chemical change is taking place? This is what called biosynthetic or biogenetic pathways. What all the enzymes which is involved in it. So that is what we are going to discuss in this class. So here in this slide it is shown very clearly, that is, despite extremely very characteristic of living organism, the pathways of generally modified synthesizing carbohydrate, proteins, fats and nucleic acids, and essentially and same to all organisms. Kingdom plantae, kingdom animal, kingdom fungi, kingdom bacteria. These processes demonstrate the fundamental unit of living matter and are collectively described as primary metabolism, with a compound involved in the pathway being termed as primary metabolites, necessary for basic survival of the organism, used for energy and the tissue construction. Even we have seen the same word in the biochemistry also. This is plant biochemistry. You can take like that. Even in the many books they used to mention that plant biochemistry includes most carbohydrates, amino acids, proteins, lipids, nucleic acid and some vitamins and cofactors. Secondary metabolites. Secondary metabolites. You can see here secondary metabolites. Secondary metabolites. So as assembly from pieces of primary metabolite, or there is an assembly of primary metabolites or plenty divert of the plant based food products, foods, and may be more prevalent and unique to certain gene species, and similar compound occurs with genus and families. Some families they can produce a particular compound, alkaloids, Solanaceae, Fabaceae, like that one families can also be construct for a certain chemical compounds and their active ingredients often have virtual function in source, attractants and propagation of species, and secondary metabolites sometimes used by the plant for attraction of some birds or something, and to make the propagation of the species they may take that to one place to other. And defensive mechanism, predator, sometimes some secondary metabolites will have some very the flavor or a smell will make the predators or the attacking insects may not come near to the particular flower or particular plant. That is also one way they used to protect them, by means of producing some secondary metabolites, signaling. This is about genetic coding, or a hybrid or any other varieties can be produced by means of this secondary metabolites are used for that, and may have useful nutrient benefit to humans and other organisms. And genes and enzymes guiding biosynthetic vary from one organism to the next. And see, this is one slide which I have taken to give a, it is taken from it is from even Trees and Events, and also you can see Brady and Taylor Pharmacognosy book also this will be having, and you can see very clearly. It's better for the students to have some reference usually, not only depending on the handout or a presentation which is presented by a teacher. You should go and refer library, utilize library always. That is very important. Library, reading books and knowing about the authors, these all are very important quality for a student to have to improve their knowledge, and it will helpful for your future study. You see this, in this I have seen side I have made in this slide saying that 4 major pathways: assembly of C skeleton, acetate, cycamate, mevalonate, DXP, that means it is deoxyxylulose phosphate. Most commonly this xylulose 5-phosphate is a precursor, always it is coming across like when we are using phosphoenol pyravate. So that is one, that is why it is also an intermediate product which comes for various metabolic pathways in plants. And cycamic acid pathway, acetate pathway, and N, that means nitrogen is needed for any amino acid. So nitration amino acid, even for alkaloids, always nitrogen containing compound is alkali, when you see the definition of alkali. So these all are secondary metabolite pathways. So here we are going to discuss about cycamic acid pathway, and this picture what I have shown here in the slide, I have made, there are many slides which is repeated, and this slide is again repeated to make it clear. You can see now the slide of the structures and the reaction in one slide little more bigger, because I just removed that 4 explanation for the 4 synthetic pathways separately, which I have mentioned little before regarding acetate, cycamate, mevalonate and DXP. See here, what is written in this slide? Top: carbon dioxide and water. In the presence of sunlight, energy, the plants can produce starch. Just we have taken an example of algae, but all the plants have the production of starch. And what happens? Glucose and other starch, and it is converted into polysaccharides, that is starch. And then you can see the glucose molecule enter into glycolysis and gives rise to phosphoenol pyravate, which you might have studied in your biochemistry. The same thing, glycolysis will give 1 molecule of glucose can give 2 molecule of pyruvic acid, that is phosphoenol pyravate. Similar, it is phosphorylated. And then this, you can see the cycle. Even the carbohydrate directly they can give pentose phosphate pathway, means acetate pathway, which can give rise to RNA and DNA nucleotides. Pentose phosphate pathway. Pentose. Pentose means that is you can say that is, it is an carbohydrate with 5 carbon atom. Understand? So that is also one pathway which can give rise to RNA and DNA, and erythrose 4-phosphate can also give rise to sikimate. See the structure of sikimate, sikimic acid. Remember, so that next slide when it come, when I was telling about this sikimic acid, you can remember this structure. You can see that there is an carboxylic acid group, a functional group in that one position, and down you can see in the third, fourth and fifth position it is OH group is present, and a single bond which is present in sikimic acid, and it is called sikimate. Why? Because it is in ionic form, or in a salt form you are telling sikimate, otherwise it can be called as sikimic acid. This basic chemistry you might have studied in your organic chemistry very clearly. When you will call say acid and when you will call salt, it is ate acid or a sikimic acid or sikimate, acetate or acetic acid, like that. Ate when you are using it is an ionic form to form an cation, or an anionic form, or a salt when it is forming you will say ATE, and it can produce ligands and coumarins. And see, even the sikimic acid can give rise to aromatic amino acid, and it can give rise to protein, enzyme and alkaloids. So this is what it is shown in this slide, and we are going to discuss only today about the sikimic acid, how it is formed and how the aromatic amino acids are formed. We will try to finish that one, and then we will continue with the next class like that. So we have to remember what is sikimic acid, what is aromatic amino acid, and how it is formed by means of this biosynthetic pathways which is happened in the plant. And next you see the pyruvate can also undergo next step. It is acetyl-CoA. Acetyl-CoA enzyme. You might have studied about acetyl-CoA. And acetyl-CoA is converted into mevalonic acid. And mevalonic acid, that is mevalonate pathway. Mevalonic acid can give rise to terpenoids, steroids, carotenoids. These all are present in the plants. Terpenoids, steroids and carotenoids. And you can see that, yes, that is, for example, acetyl-CoA enzyme can also combine with fatty acid polyketides, and it can give rise to polyphenols, prostaglandins, that is macrocycle X antibiotic or macrolytic antibiotic. And also you can see the tricyclic acid, citric acid cycle or TCA cycle will enter into the acetyl-CoA enzyme into the TCA cycle, and then you can have aliphatic amino acids also. All the aliphatic amino acid present in the nature can also produced by means of from the TCA cycle, and you can get proteins, enzymes and alkaloids by this whole picture. So you can see a grand idea. A student now got one idea what we are going to discuss in this class. We are going to discuss about basic metabolic pathways to produce secondary metabolites from a primary metabolite in plants. So what is the primary metabolite mostly present in the plant? Is a carbohydrate, a polysaccharide, a starch, and it can undergo into various pathways by means of taking this advantage of the carbohydrate which is present as a primary metabolite survival of the plant, so that it can be used by the plant by means of various reactions. These reactions which is taking place naturally in the plants are said to be biosynthetic pathways or biogenetic pathways. So here you see the metabolic pathways of sikimic acid and aromatic amino acid and phenolic compounds which we are going to discuss according to the syllabus. And shikimic acid or shikimate is an important biochemical metabolite in the plants and microorganism. And aromatic compounds is a hydrocarbon with altering double bonds in a single bond between carbon atom. Amino acids are molecules containing an amine group, a carboxylic acid group as a side chain that every student may be knowing. What is an amino acid? Amino and acid. Amino group and an acid group will be present in an amino acid. And also they are called as deuterions because of presence of they have anionic character and cationic character in the same molecule. That is why they are called as deuterions. These all things you might have studied in your smaller classes. So you should remember all your basic chemistry because we are coming to the high version of the chemistry. So you need the basic chemistry. If you are not remembering, go and read your previous handouts and notes which were given by your teachers. Phenolic compounds are large and diverse group of molecules which includes many families of aromatic secondary metabolites in plants. Enzymes are group of molecules that serve as a catalyst with a high degree of specificity for a certain substrate or a class of substrate. It can only act as one substrate or on a family of structurally similar substrates. Major enzymes which we are coming across in this study is synthetase. What it does, joins two molecules together without hydrolyzing, with or without a pyrophosphate bond. And you can see dehydratase removes water to create a double bond. Dehydrogenase removes hydrogen atom from its substrate. Kinase transfer a phosphate group from a high energy phosphate compound such as ATP to its substrate. And this slide shows that shikimic acid, it is you are telling almost all the plant, if they have a secondary metabolite, it is an aromatic compound, it needs us to pass through shikimic acid. And even the shikimic acid alone is also present in certain plant, and this is the source of the plant where it is present. It is Shikimi Elysium anisatum. And this one anisatum is a flavoring agent which we might have seen in various non-veg preparation in our home. Even in biryanis we might have added this one, and it contains shikimic acid. And in this slide, students should see very clearly what is the structure of the shikimic acid. Because you have come across all your basic chemistry, it is needed for the students should know what is this by means of. I did not put everywhere CH2, CH or something like that. It's a basic chemistry. If you know, you can understand. It is a 6-membered ring structure which has a double bond inside, and there is 3 OH groups are there, and there is COOH group is there in the top. So you can see there is an acid group is there. That is why it is called shikimic acid. Acid group is in the 1 position, and then you can see all the 3, 3rd, 4th and 5th position, they are having OH group. If it is there a double bond and which 3 OH group in a 5-membered ring structure with a carboxylic acid functional group in the 1 position, then it can be called as shikimic acid. If you remember by means of orally, it is possible for you to write in the paper the structure. And next you can see shikimic acid is a precursor for verol. It is precursor for aromatic amino acid, phenylalanine and tyrosine. So when you know about phenylalanine, immediately you should remember about phenylalanine and tyrosine structure should come into your mind immediately. Do you remember phenyl? Phenyl means it is a benzene group. Am I right? And benzene. Phenol means it is having OH group. Phenyl. Alanine. Alanine is a is an aliphatic amino acid. Initially you might have studied aliphatic amino acid, which contain one amino group and one acid group and one CH2 CH2 group will be there. That is alanine. So when alanine is attached with a benzene, then it is phenylalanine. Then what is tyrosine? Tyrosine is coming from the phenylalanine, just one para position. In the fourth position, if you have one OH group extra from phenylalanine, then immediately it is called tyrosine. Without showing the structure, I am giving orally, a person can remember what is phenylalanine, and what is the tyrosine chemical structure can be remembered in your imaginary or in your imaginary picture, in your imaginary screen in your mind. It is possible, it is visible for you. That makes the student to understand. In every chemical study, when you are chemistry, when you are study, always you should bring it in your picture. It should bring it in your mind. What is its structure? I hope you followed me. And what is indole? An indole derivative. Indole means what? What do you mean by indole? Indole structure. You have studied this in your heterocyclic compounds, in your organic chemistry, in your POC, you in last semester, you should have studied heterocyclic compound. In that indole, what do you mean by indole? Indole means it is having a 6-membered ring and a 5-membered ring with one nitrogen. That is the indole structure. So that is why an indole derivative, tryptophan, will have one 6-membered ring and a 5-membered ring structure with nitrogen. NH will be there, and it will be having a side chain with an amino group, and that is another, it is acid group, CH2 COOH will be there, and that is called a tryptophan. What is the AAA stands for? Aromatic amino acid, simply a short form. I have just written there. Aromatic amino acid tryptophan. An aromatic amino acid phenylalanine, aromatic amino acid tyrosine, and alkaloids also, it can be produced by succinic acid. Why? Because alkaloid also, there are many alkaloids which is having aromatic ring. Wherever there is an aromatic ring, it needs to pass through succinic acid cycle or succinic acid pathway. So succinic acid pathway or succinic acid plays as a precursor for production of alkaloids in the plants. And phenyl propanoid flavonoids, and propanoids, flavonoids, and tannins and lignins are also having aromaticity, and that structure also produced by this precursor succinic acid. So next slide, which you are coming to the formation of succinic acid. How you are going to form a succinic acid. So succinic acid, you can see pyruvate is converted into phosphoenol pyruvate. See the structure, pyruvic acid. It is C double bond O, and that is COOH, and the C is connected with phosphate, and then you are getting C double bond CH2. That is the phosphoenol pyruvate. A pyruvic acid can be converted into phosphoenol pyruvate in the conversion of ATP to ADP. A triphosphate is converted into diphosphate, and this is the starting material of a reaction succinic acid, how it is formed. Now you should know the structure of 4-erythrophosphate. So initial, always whenever you are producing some compound, even in the synthetic way in the laboratory, we will be having some reactant. So here we have taken a reactant. One is pyruvic acid. We have not taken, it happens naturally in the plant. We are studying that. That point you have to remember. If I say mistake also, it is responsibility of the student to know that, okay, he's telling about the plant's living organism. It is a biosynthetic pathway, you have to remember. So you see, the pyruvic acid and this erythrose 4-phosphate. How many carbon atoms are present in pyruvic acid? It has 3 carbon atoms. How many carbon atom is present in this slide which I am showing, that erythrose 4-phosphate? It has 4 carbon atom. So when these two join by means of an enzymatic action, how many number of carbon atom will be produced in the product, in the intermediate next product? It will be having 7 carbon atom. So 7 carbon atom means what will be the name? If it is a carbohydrate, it should be a carbohydrate. So what will be the name? It will be heptulose. You might have studied triose, tetrose, pentose, hexose, heptose, octose, nanos, like that you might have studied. So this is heptose. Heptulose, 7 carbon atom compound. So you see this picture, see this, there will be many pictures repeated will be there, understand the structure. So just to make the student to study, it's only the number of slides will be more, but the content of the lesson will be very less, because the student should be clear, then only they can write in the structures in your exam. So you see here, I have written in another form. Another form. There is in the top, it is pyruvate, phosphoenol pyruvate is combined with erythrose 4-phosphate, and you can see in the reaction, that is phosphate, removal of phosphate, one molecule, and immediately joining the 2, 3 and 4 carbon atom compound is converted into 7 carbon atom. Can you see that? If you count 1, 2, 3, 4, 5, 6, 7. 7 carbon atoms are present. It is an heptulose compound. And this heptulose compound is converted into, by means of this nucleotide adenine dinucleotide catalyst. It is a catalyst or cofactor. By means of this cofactor change, immediately you are getting a cyclization. What is the name of that compound? It is not sechemic acid. Sechemic acid means, from the beginning I was telling, it should have one COOH in the number 1 position, and 3, 4, 5 you should have OH. This is not having a 3, 5, 4 OH group, and there is an extra OH is also there, a double bond O, and that is, this is not sechemic, this is cunate, and again it has to be converted. But anyhow, it is cyclized and it is forming first one compound, and next it is having next 2, 3 reactions. After that only you are getting sechemic acid. That every student should remember. But it is the form of sechemic acid. Understand? So now you can see what is the enzyme responsible. We are going to discuss about the enzymes. So 3-dehydroxy D-arabino heptulosynate, because you want a 7 carbon atom compound, 7 phosphate synthetase, that name. So always it is easy for you to study the enzyme name. Always what is the product, if you know, you can make the enzyme name. Understand? Synthetase, it synthesize a 7-membered carbon atom from the 3-membered carbon atom phosphoenol pyruvate and 4-membered erythrose 4-phosphate joined together, and in the presence of 3-dehydroxy D-arabino heptulosynate 7-phosphate enzyme is the first enzyme which will give you the reaction in the sechemic acid pathway. By this enzyme directly it is, you are not getting sechemic. There are many other enzymes also there. Since it is the first enzyme of the sechemic pathway, it controls the amount of carbon entering. It controls the carbon entering pathway. It is the deciding factor for the number of atoms which is joining carbon. Formation of sechemic acid. See, this is again the same explanation is repetition. You can see there erythrose 4-phosphate plus this phosphoenol pyrophosphate in the presence of the enzyme DAHPS. What do you mean by DAHPS? You can see the deoxy arabino heptulosynate phosphate synthetase. Yes, is added. This is for product name D3-deoxy D-arabino heptulosynate 7-phosphate. So very simple. Same name. Only in a short form it is written. Can you follow me? So that is very easy to write. And you see, this is the full form, the same thing which you are studied in chemistry. You might have studied the reactions and all. So just I repeat it, and to show you and to remember, to clearly the student should remember the structure of the compound. Now also the student will remember now. There is erythrose 4-phosphate, there is a phosphoenol pyruvate. These two molecules will give a 7-membered structure. See here, in this structure, it is phosphoenol pyruvate, combine with erythrose 4-phosphate and give rise to 7-membered compound, and then it cyclizes and it gives this one. I can see that double bond O, one OH it is there and another OH is there. So it is called cunate, and cunate is converted into another compound. It is nearing to succinic acid, but it is not succinic acid. And what is the name of that? Dehydro. It can be called as 3-dehydrosuccinic acid because only in the third position double bond O. All the other are okay. And then when you NADPH, that is when you NADPH cofactor acting and gives succinic acid. So this anyone can remember, please. Once, one or two times when you see the slide or listen and remember and memorize so that you can remember. Then you can write and see. Always chemistry you have to write and see. By vision or listening may not give you much practice for writing. The examination time it will be difficult. So better you can write and see what, how and the structure is formed. So here you have to remember, after this 7 heptylose or 7-membered carbon atom compound formation, you are having two intermediate products. You have to remember cunic acid, then 3-dehydrosuccinic acid, and then it comes converted into succinic acid. And so now you see 3-dehydrocunate synthetase. This is what I told. Next, first one is cunic acid. So 7-membered carbon atom is now converted into dehydrocunate or 3-dehydrocunic synthetase is used. A second enzyme of the succinic acid pathway. It catalyzes the elimination of phosphate from the dehydroarabinoheptylophosphate to generate the dehydrocunate. And you can see here, this is in structure wise I have shown. 3-dehydroarabinoheptylosinate 7-phosphate is converted into dehydrocunate by means of the enzyme dehydrocunate synthetase. So the enzyme is very simple. You can just write, and the cofactor NAD is converted into NADH. And you can see 3-dehydrocunate dehydrogenase is catalyzed by third step to cunate pathway, that is 3-dehydrocunate to 3-dehydrosuccimate. So see here the same thing is again we have made into a short reaction, that is 3-dehydrocunate is converted into 3-dehydrosuccimate. Now 3-dehydrosuccimate have to convert it into succinic acid because you know that succinic acid structure all 3, 4, 5 should be OH, and in the first there will be acid group. So you can see succimate 5-dehydrogenase. The fourth step of succimate pathway is reduction of dehydrosuccimate to succimate. The reaction is catalyzed by NADP dependent and succimate dehydrogenase. And you see this one formation of succinic acid, as I told, 3-dehydrogenase succimate is converted into directly D-succimate or succinic acid. Succimate we are saying because it in ionic form the structure is written. And say next step again the succimate kinase, because we want to go to the next step. Succinic acid we have formed, we are going to study how the aromaticity is formed. Aromatic compounds are formed. How the aromatic means you need a benzene with 3 double bonds. Succinic acid is having only single bond. So it is not even still, it is a cyclic compound, but aromaticity is not achieved. So how to achieve this aromatic compound? The next step. In a fifth step of the succinic acid pathway, succimate kinase catalyze the specific phosphorylation of 3-hydroxy group of D-succimate to yield succimate 3-phosphate using ATP as a cofactor. So you can see this D-succimate is converted into succimate 3-phosphate by means of succimate kinase. And 5-enol pyrovyl succimate 3-phosphate synthetase is the enzyme. in the sixth enzyme of the succinic acid pathway. It catalyzed by reversible formation of 5-enol pyravyl succimate 3-phosphate from succimate to 3-phosphate and PEG. That means you can see that is the phenyl, that is erythrose phosphate. So you can see that is erythrose phosphate, or you can see phenyl. That is you can see that is enol pyravate succimate with phosphate. That is what you can see. That is short form I have written, phenyl, or you can say PEG, and PEG will be formed in the next format, but still PEG again the student should not, even I was unable to clearly explain that is PEG. We are telling that phosphoenol pyravate, that is what we want to PEG, phosphoenol pyravate, that is what we want to show you. So you can see phosphoenol pyravate from succimate 3-phosphate and a phosphoenol pyravate. So you see, better you can note it down, PEP, PEG, not PEG, PEP is shows that phosphoenol pyravate. So phosphoenol pyravate is also needed to be added with this succinic acid 3-phosphate, which we got from the kinase enzyme reaction. We are getting the next product 5-enol pyravyl succinate, or succimate, not succinate, succimate 3-phosphate. So you see this one, next step, the succinic acid pathway is now going to the next step. So after succinic acid, what is the necessary for you to remember? You are adding a pyravate, a pyravic acid, by means of it is attaching in that particular position of this fifth position, and next you are getting corosmate synthetase. You are adding a corosmate. So how you are going to form the seventh and the final step to make a corosmate, a trunk of the succinic acid pathway, and transfer and elimination? There is a two types of reaction takes place, and it is also a conversion of carbon atom changing is there. So it is a very, very important reaction takes place. It is not directly getting from the succinic acid to corosmic acid or corosmate. Succinic acid and corosmate, how it is formed? You are adding phosphoenol pyravate and a compound, and a pathway is getting yield of corosmate. You can see this in this reaction. The second and the third double bond of a benzene ring is introduced. Our aim is to get an aromaticity. So second and third bonds are introduced in this reaction. This reaction is catalyzed by corosmate synthetase and requires reduced flavin for activity even through the overall reaction of redox neutral. And so you can see that is 5-enol pyravyl succinate 3-phosphate is converted into corosmate. So two double bonds we have achieved, and but still we have to come to the aromaticity by means of the seventh step. So seventh step of succinic pathway links the metabolism of carbohydrate to biosynthesis of aromatic amino acid and many aromatic secondary metabolites, including tetrahydrofolate and ubiquinone. That is, other products can also be produced in between. So see here in this, it's a overall structures which I have made the cycle, see, which we have discussed now. We have discussed just now about this, about all these things. So from starting from phosphoenol pyravate, it is addition of erythrose 4-phosphate. It is converted into 7-membered compound heptylate, and the enzyme which is responsible you have to remember. And then you are getting a synthetase will give you cunate synthetase. It will give cunate, and then you are dehydrogenase. When you are using, you are getting 3-dehydro succimate, and then you are using a dehydrogenase of succimate will give succinic acid. And succimate is using by means of kinase phosphorylated, and then this one is added with an phosphoenol pyravate, and then you are getting 5-enol pyravate succimate, which is converted into corosmate by means of corosmate synthetase, and there you have got two double bonds in corosmate. And you see this is the same the explanation which I have given there. This is only a colored structures which is given, and same thing you can see phosphoenol pyruvate with erythrose 4-phosphate in the presence of the enzyme. You can see enzyme will be the same products enzyme, heptulose enzyme, and then you can see there is cunate synthetase enzyme, and then you can see that cunate enzyme and succinate dehydrogenase enzyme, kinase enzyme, how it gives rise to the and you have to add this phosphoenol pyruvate, that you have to remember, that is PEP. That PEP which I mentioned here, you have to remember that is phosphoenol pyruvate, where we stuck in one slide. Do you remember? Before I just stuck and I was going behind and coming back. So that will make you to understand where my teacher also got a doubt about PEP. Phosphoenol pyruvate you have to add, then only you are getting 3-enol pyruvate succinate 3-phosphate, and then you are getting a corosmate is formed. After corosmate, still corosmate is having only 2 bonds. So next step we will think about how the corosmate is converted into the next one after knowing about aromatic amino acid. Here in aromatic amino acid you see only 3 we are going to discuss about aromatic amino acid. I have shown 3 pictures. In that you can see that is 3 pictures which I have shown in this. You can see F, Y and W. I have given some symbol. So what is the difference in these 3 and what are they? By means of the structure itself you should identify your mind what is the structure you have. See, F is having 2 red color. Red color points are denotes in the chemistry. We can see that is an O, oxygen. That is, one is OH, another one is double bond O. So here one F is having one double bond O and one OH. So you can see this is a compound which is having a benzene ring with CH2, and there is one double bond O and OH. So an acid and also one nitrogen, that is amino group, is also present by means of a blue color. So you can see this is phenylalanine. And you can see here in the Y shape there is one red color in the 4th position, and below the benzene ring also having one red color which is OH, and you can see in the top same structure which is shown in the F. You can have there is one structure which is having, see, there is a power have gone, so we were unable to have one light. Anyhow, I'm just continuing in this recording by means of just removing this window to get light, and so that we can continue our audio. The power have gone. So anyhow, during recording I don't want to stop this recording. This is by natural light also. You can I can record this and then I will upload. So you can see here this is the structure of as we have discussed. The first one is phenylalanine, and Y is having one OH group, so it is tyrosine. And you see W, there is 2 rings are there. So one is 5-membered ring, another one 5-membered ring is attached with the 6-membered ring of aromatic compound. So, and there is one nitrogen is also present in the 5-membered ring. There is a blue color, and so that it is indole, an indole derivative which is containing one nitrogen NH2 group, and also it has COOH group also is there. So you can see it is also tryptophan. So we are going to see the structure of aromatic amino acids, how it is formed by means of this reaction. So aromatic amino acid, what are aromatic amino acids? Aromatic amino acids are amino acids that include an aromatic ring. For example, includes phenylalanine, tryptophan, histidine and tyrosine. So as we have seen in the slide before, what we have discussed about F, W and Y, you can see F stands for F. I have showed that is it has no OH group in the 4th position, so it is phenylalanine. W, we have one OH group in the 4th position and an alanine in the 1st position, so it is an tyrosine. And Y, we have the structure of the Y we discussed, that is it has a 5-membered ring and a 6-membered ring. A 5-membered ring has a nitrogen and also an attachment with NH2 and COOH group in that attachment in the substitute. So it can be the main, that is indole derivative, that is tryptophan. So histidine, we are not going to study this one, but histidine also comes in the aromatic amino acid, natural aromatic amino acid. So here in the plant and microorganism, phenylalanine, tyrosine and tryptophan are the common, and also the precursor which we need is, that is phosphoenol pyruvate, erythrose 4-phosphate and corosmate, which is very much needed for these factors. And you can see the starting material is corosmate, which we have discussed previously. So corosmate will be there, and this what all the enzymes which is responsible for these reactions are isomerase, mutase and transferase. Isomerase is an enzyme that catalyzes the structure rearrangement of isomers. Mutase catalyzes the shifting of a functional group from one position to another with the same molecule. Transferase catalyzes the transfer of a functional group, methyl or phosphate, from one molecule to another. And you can see this structure which I have shown, this slide. You can see this slide. In this slide, you can see that is succinic acid is how it is converted into a side chain with the help of a phosphoenol pyruvate, and then it is converted into andronic acid. Andronic acid will give rise to tryptophan, because andronic acid is the main acid which can give rise to the 5-membered ring structure. But in the same time, the other structure, you can see corosmate mutase, or the variant enzyme will be given, you will give rise to a compound which will give rise to phenylalanine and tyrosine. So these 3 aromatic amino acids can be formed from the succinic acid in this structure. And also you can see the mechanism. Corosmate will be converted into prephenate. What is prephenate? Prephenate, you can see this is the rearrangement of prephenate. Very important how it can be changed by means of, only by means of a scientist he has identified, it is by Clenson rearrangement, because there is a big rearrangement takes place. In this reaction you may not be very clear. You can see in the next slide, which first you should know what is a Clenson rearrangement. That is Claisen. That is Claisen rearrangement. I told Clenson. So it is a Claisen rearrangement. The Claisen rearrangement is a powerful carbon-carbon bond-forming chemical reaction discovered by Rainer Lewin Claisen. And you see, what is that reaction? This is the reaction. So you see here, corosmate is, you may be knowing very clearly, there is in the third position you are having an attachment by means of phosphoenol pyruvate and an acid, and then you can having a shift from the third position to the first position. Not only that, even a shifting of carbon atom also takes place. So you see that is CH2COO, which is converted into CH2C double bond O COO, and the position is also shifted. So that is what prephenate is formed from corosmate. And for phenylalanine biosynthesis also, you can see phenylalanine biosynthesis very clearly by the structure which is shown is here. It is an prephenate is converted into phenylalanine. Phenylalanine in a benzene structure with 3 double bonds and one CH2C double bond O COOH. That is the one alanine group, which is actually, it is not phenylalanine, it is phenyl pyruvate. Why it is not called phenylalanine? Because alanine means it needs NH2 group. There is no NH2 group in this slide which I am shown. So you have to remember, student, this is not phenylalanine, this is phenyl pyruvate. So how it will be converted? Now the phenyl pyruvate is converted into phenylalanine by means of using one amino transferase enzyme. So an amino group is added. You can see in the final product structure, there is a benzene with CH2CH NH2 COOH is mentioned in the last structure, which gives the clear picture of phenylalanine. And next one is, this phenylalanine is present in various milks and even pulse, and also it is present in wheat and beans. It is present essential amino acid. And tyrosine tyrosine is just one OH group will be present in the phenylalanine. So prephenate can be converted by means of NaNADH directly to 4-hydroxy phenyl pyruvate, and then if you do animation, then amino group is added. when you add amino group to that particular structure, you will get tyrosine. See here, the same thing. Only thing is you are taking that amino OH group with prephenate, and then you are making it, you are getting an hydroxy, that is para-hydroxy phenyl pyruvate to is you are obtained, and then you are using amination can be done by amino group. When you added in that double bond you are getting tyrosine. Tyrosine is also present in banana, chicken, and various, even avocado and even almond, or all it is an also essential aromatic amino acid. Overall, when you want to see tyrosine and tryptophan, very simple. Corosmate is converted into prephenate, or chlorosmic acid is converted into prefemic acid, and prefenic acid is converted into phenyl pyruvate, and you are getting phenylalanine. Here 4-hydroxyphenyl pyruvate, you are getting tyrosine. So this is you have to remember. Next you can see tryptophan. As we want to discuss about tryptophan, it is also corosmate to anthranilate, anthranic acid. This reaction is catalyzed by anthranilate synthesis. Anthranilate N5 phosphoribosyl anthranilate. This reaction is catalyzed by anthranilate phosphoryl ribosyl transferase, and N5 phosphoribosyl anthranilate is enol 1 carboxyl phenyl amino and 1-deoxyribose phosphate. This reaction is catalyzed by that is phosphoribosyl anthranilate isomerase, and then the enol 1-O carboxyphenyl amino, 1-deoxyribose phosphate, and 1-indole 3 glyceryl phosphate. This reaction is catalyzed by indole 3 glyceryl phosphate synthesis, and indole 3 glyceryl phosphate to tryptophan. This reaction is catalyzed by tryptophan synthesis. And overall, if you see this reaction, indole, that is for example indole derivative tryptophan can be, that is it can be corosmate, can be produced, converted into anthranilate. Anthranilate is converted into phosphoribosyl anthranilate, and then it is converted into enol 1 ortho carboxyphenol amino deoxyribose phosphate, and then it is converted into and tryptophan by means of tryptophan synthesis. So indole glyceryl phosphate is an intermediate between this compound, and then you have to remember the synthetase enzyme which is responsible to give you tryptophan. So tryptophan is also most of the common, it is present in the algae and eggs and fishes is also present, very coccolades also it is present. So today's class we are ending with this tryptophan, and also how we are that is how you are used this ischemic acid pathway for the production of amino acid, and then the aromatic amino acid. So this will be, I hope this will be helpful for you, the students who have attended on 16th July 2020, and also you can have the Thursday. If any audio problem or anything, you just listen once more and you can have this uploaded lecture. Thank you. We will see in the next class about the pharmacognosy.